Namaskar dear students, welcome you to our channel Bio by Gulab sir. As a part of series of lectures, today's topic is blood group. It is very popular topic, that blood group, and very interesting to know about our blood group. So very often you used to see and we used to ask each other, what is your blood group? So how to know about that blood group and what is blood group, that is what I am going to explain you. Here there are four major types of blood group which are in syllabus also, and they are common blood group except Bombay blood group and blood group M and N. So we are going to learn about this first common blood group. See, there are four blood group A, B, AB and O. How to know or how they are recognized or how they are produced in our body, why they are so called as blood group A, B, AB and O. Blood group A is because of that special antigen present over RBCs. It is produced by the genes present in our body. The genes IA or small I. That is what in homozygous or heterozygous condition, A is being dominant. It is active gene producing antigen A and it get deposited over RBC. This antigen A is special type of glycoprotein. That glycoprotein A gives blood group A. Same way for blood group B, having genotype IB or IB small I. That genes produces a special antigen glycoprotein that is of B type and it is over RBC. Therefore, that person who has that glycoprotein B over RBC is of B blood group, or that person has B blood group. Now what is AB blood group? So person has genotype IB. That being both dominant and co-dominant gene, they undergo transcription and translation to produce say both antigen A and antigen B, and that both antigen A and antigen B are present over RBC, and therefore the blood group of that individual is AB. And finally the O blood group, because of recessive gene small I, they do not produce any antigen and therefore there is no antigen over RBC. So that blood group is recognized as O blood group. To know more about this blood group, that what kind of antibodies are there, because on the basis of that presence of antibodies, that donor and receiver is being decided, and you are known that who is the donor and who is the receiver. Moreover, who is the universal donor and who is the universal receiver. As we know that O blood group is always called as universal donor and AB blood group is called as universal acceptor or receiver. Why? That is what let know. Here antibody B is present in blood group A, and therefore person with blood group B cannot donate blood group to the A since it has antibody B. That means this antibody B destroy the antigen and RBCs of person with blood group B when it is given to blood group A. Means that is what antibody B fight against antigen B, and therefore blood group with B cannot donate to the blood group with A. Same way, blood group with A cannot donate to the person one who has blood group B, because that blood group B has antibody A, that is against A, antigen A, and therefore if this blood group B is given, or person with blood group B, that blood is given to the person with blood group A, this antibody B will destroy this antigen over B, and if the blood group person with blood group A, that blood is given to the person with blood group B has antibody A, will destroy antigen A, means clumping will occur. Therefore these two persons cannot give blood group to each other. Now see, in case of AB has no antibody, therefore if blood group A is given or B is given or O is given, that there is no question of clumping. Therefore that blood group AB is receiver because no antibody, and therefore if blood come from A or B will not have any clumping or any agglutination, and that person will not have any problem. Now in case of O can donate because it has no antigen. It can donate to A, AB, B and A, and therefore it is called as universal donor. Why? Because no antigen. But blood group O has antibody both A and B. Therefore blood from A and blood from B or blood from AB, if given to the O, will cause clumping or agglutination, and that individual will die. And therefore person with blood group O cannot receive blood from AB only and only can receive blood from O blood group person. And therefore it is very clear to you now that donation of blood is depend upon the antibodies present in that person. And therefore A can give to the A or O can give to the O. B can give to the B or O can give to the B. AB can receive but cannot give to A or B, because AB has both antigen A and B, and A and B has antibody A and B. So person with blood group AB, if it is given to either A or B, will have a clumping. That you got it, and it is very clear to you now. Here second point comes, if we understand what blood group A, B, AB and O are, then what is about positive and negative blood group? Any person with any blood group, either A, B, AB or O, if has rhesus factor, that is first time found in rhesus monkey, and therefore the name given rhesus factor. So the gene with dominance produce that rhesus factor which is over the RBC. If it is present, it is called positive. If it is absent, it is called negative. So blood group A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative, and these are the genotypes what I told that control the formation of that specific antigen being present in dominant form. If it is recessive, no production of antigen, and therefore blood group O has no antigen over RBC, and therefore it is O. I hope you got it, these four major types of blood groups. Now let us know how this blood group can be checked, to know how to know this blood group, how that laboratory technicians recognize the blood group when you give your drop of blood or blood. So they test your blood and say what is your blood group. Now see what I have drawn. Let you know first what technique is. This what A, B, D I wrote are the antisera. That means that are the antibody, antisera. And these 3 drops of blood on each slide what I have shown. Suppose I have to test a person who has blood group A, we have taken that 3 drops of blood from that person. In that 3 drops of blood, we have to put 1 or 2 drops of antisera A, antisera B and antisera D separately. Now see, as I told you, antisera A is antibody A, and therefore if blood group A is having that blood, if we add antibody A, we will have clumping, and that clump मतलब जैसे बूंदी बूंदी जैसा आपको उसमें एक्सप्रेशन मिलेगा, and वो बूंदी जैसा एक्सप्रेशन will gives us indication that this blood group is A, and if clumping occur in antisera D, that is for rhesus factor, then it is A positive. If clumping occur only in A and not in D, B they will not occur, because question is about blood group A, therefore there is no question to test about B. So A negative. So in case of B, clumping will occur in B because antisera B is added and there is antigen B. So clumping between antibody B and antigen B. There is no clumping with A, and therefore the blood group will be B. If D added and clumping occur, this is B positive. Here in case clumping occur in B case but not in RH, therefore is B negative. And the same way for AB. Now see, AB has both antigen. When antisera A is added, antisera B is added, in both the case it will have clumping that indicate it is blood group AB, and in case of RH factor, if clumping occur, it is positive. Means in case of AB positive blood group, clumping will occur in all these 3 drops. Now if it is negative, no doubt, in A and B case, clumping will occur, but RH positive case, RH rhesus factor case, it will be negative, that is AB negative. Now see that very interesting about O blood group, that O, since has recessive genes, will not have antigen A and B. If antisera A is added and B is added, since there is no antigen, there will not be clumping. That means that is O positive with RH factor. And in case of O negative, clumping will not occur in anyone. Since no antigen A, no antigen B, no RH factor, and we've added this antisera A, antisera B, antisera D, will not have clumping, that indicate it is O negative. And very important to know and say to you that O negative is always ready to donate to anyone who got accident. That means at the time of accident, if that person immediately need blood, then there is no question of, or there is no time to check the blood group and donate the blood group. By that time, blood group available in blood bank O negative is immediately served to donate to anyone one who required the blood. Why? Because no antigen A, no antigen B, no rhesus factor, no question. That one who received the blood. Therefore, it question can be as a NEET. One of the following blood group is always called as a ready blood group to donate anyone is O negative. If assertion and reason question asked, that is what O negative blood group is always ready to donate to everyone, because there is no clumping in the person either with positive or negative blood group. Why there is no clumping? Because there is no antigen A, antigen B or rhesus factor if it is given to the person with blood group A, B or AB or O. Why? There is no antigen. If that person has antibody, even though there will not be clumping, and therefore it is called ready blood group to donate. I hope you got it about this, how to know our blood group. You can refer these notes, same thing I have written here, what I explained to you. Now let us know about very interesting blood group, that is called Bombay blood group, which is not in syllabus, but you would have heard the word Bombay blood group. What is it? It is very rare blood group, and accidentally it is being observed by Dr. Y.M. Binde. Dr. Y.M. Binde, he observed in Bombay. What he observed? A person who has genotype IA, IB or IB, that this person must have either blood group A, AB or B. But instead of or in spite of having genotype IA, IB or IA or IB, they don't have the blood group A, B, AB. Matlab genotype with IA must have A blood group, but it is not found in that person's because it is not tested for blood group A. Why? Let us understand. To produce blood group A, B and AB required a special protein which acts as a precursor, and it is produced by a gene. That gene is called gene Ch-18, that is F51, which produces that antigen H is a precursor of A. If that is being produced, then only that IA genotype which produce enzyme catalyze the process to convert that precursor A into antigen A. Otherwise, if it is not present and that IAI is present, the enzyme is there but this precursor is not there, then there will not be a question to form the antigen A. In the same case, in case of B blood group, that gene Q1 produces antigen H is required for both. It acts as a precursor B, so that this gene IB produces enzyme catalyze the process to convert that precursor B into antigen B. But if that precursor B is not present, then how antigen B will produce even if that genotypes are IB are there? Therefore, it is clear that although that individual has this gene in dominant form for A and B blood group, but they do not have blood group A and B. Why? Because there is no their precursor. That precursor is supposed to convert into respective blood group antigen A, antigen B. Therefore, although the person or in spite of that person have that genotype IA, IB will not have blood group A and B because the precursor which required that is not present. That means this gene is present in recessive form, could not produce antigen H which act as a precursor A and antigen H which act as a precursor B. Therefore, that individual will have no blood group A and B will have blood group O. That is called Bombay blood group. Once again, to know the definition of Bombay blood group, although an individual has genotype IA or IB will not have blood group A and B because of that precursor A and precursor B is absent because of that recessive gene H small h which I have written. This precursor produced from future to one gene which if it present in dominant form either HH or H capital H small h, homozygous or heterozygous condition. If it is present in recessive form small h, this precursor will not produce and blood group AB will not appear an individual although they have genotype IA and IB. I hope you got it. You can refer it again as notes are always attached with our video. Now to know about something best, that is what Rh factor and Rh negative factors are always a topic of interest to know about what happens with an individual which has Rh positive or Rh negative blood, especially what happens with the female who has Rh negative blood group. That one is a female, if she has Rh negative blood group, then what problem occur in the second pregnancy and how that problem occur. Let us know about it. A female with blood group or mother with female blood group Rh negative. She has no antigen Rh. Who got married with a person who is having Rh positive blood group means for that baby father is Rh positive. So naturally the baby will have Rh positive blood group. Now mother has Rh negative, fetus has Rh positive. For the first pregnancy everything goes smoothly. First baby take the birth, but by the time of taking birth, this tissue from here this exchange of nutrition and waste material or gases occur. It may injure and this Rh positive antigen may enter into the mother body. When that Rh positive antigen enter into the mother body, by the time second pregnancy may occur, during that period that anti-Rh antibodies produced in mother body in response to that Rh factor. Why? Because Rh factor is a foreign material for mother as an antigen. In response, mother's body produces anti-Rh antibody. That means anti-Rh antibody fight against Rh factor. So by the time when second issue, that pregnancy occur, sufficient amount of anti-Rh antibodies are produced in mother body, which transfer to the fetus body who has got Rh positive blood group. So that anti-Rh antibody fight against Rh, anti-Rh, मतलब Rh के खिलाफ antibody to fight, and therefore they destroy that Rh. That means it causes the clumping. When that anti-Rh antibody enter into the fetus body, it causes the clumping, and that clumping result into the death of the baby. And death of baby is called, because of that, if death of the baby occurs, is called erythroblastosis fetalis. Therefore, with this study, you student can advise to all female members in the society, if she has Rh negative blood group, she has to marry with an individual with Rh negative. And if she get married with Rh positive blood group, then second child will have problem. And it has to be in presence of doctor. If that pregnancy is occur, it should be assisted pregnancy, that is by assisted reproductive technology. It has to be assisted by doctor. It has to be under care of doctor. Otherwise, there may be chances to have erythroblastosis fetalis. So I hope this is very interesting and very important topic that you learn what happens if the female with Rh negative blood group. Also, you learn about Bombay blood group, and moreover, you also learn about how to know our blood groups, and that was really interesting to know about. You can do this practical by having this anti-sera at your home. You can have these 3 drops of blood, take on slide, put that anti-sera, and see clumping occur in either first, second, or third case. So if occur in first case and third case, your blood group is A positive. Yes, you enjoyed today's video. I am very happy you are visiting our channel, liking us, as well as subscribing and sharing our channel. Also, I appeal you to press bell icon to receive new notification as and when the videos are uploaded. Thank you. Enjoy the learning.